Welcome, everybody. On behalf of the Center for Brain Health, this is our most popular speaker series of the year, the Brain and Owner's Guide, which we do every February. I'm Dr. Dan Krosik, Deputy Director at the Center and a Professor of Cognitive Neuroscience at UT Dallas. Before we get started, I'd like to share a little about who we are and our focus at the Center for Brain Health. We are a cognitive neuroscience research center exploring human performance, behavior, and brain function, all with an eye toward discovering how we can improve ourselves, our brains, and our lives. We are part of the University of Texas at Dallas, and we've dedicated the past 3 decades to discovering and championing a new approach in neuroscience, uncovering our brain's ability to strengthen and improve based on how we use our brains across the lifespan. The true power of research lies in how we direct it toward making the world a better place. That translation into real-world applications that benefit people and organizations is a crucial part of what we do. We've recently launched a technology platform that will allow people everywhere to become more proactive about their brain health and performance. It's called the Brain Health Project. We'd like to share a brief video on that.
We work, we live, we innovate and create. At the center of it all is your brain health. The ability to solve problems, think analytically, share empathy, and thrive.
We're trying to make brain performance really the next fitness revolution.
So how do you boost brain power? Welcome to the Brain Health Project, an urgent call to transform your mind to work stronger and faster.
This is an absolute crisis, as great as any we have ever faced. We have to equip the minds and brains of our citizens to cope with the accelerating, dizzying rate of change that they face in their lives.
Your brain health is not fixed. Scientific discoveries prove it can adapt and grow regardless of your starting point.
Our greatest value, the asset that will help us to change everything, every problem we're in, is all in our head.
To harness that treasure, we must measure and monitor progress while things are going well versus waiting for an injury or disease to strike.
Too many of us are outliving our brains, and that does not have to be the case.
The information age is bombarding us with more content than our human brains can handle. How do you keep from getting lost in this? And focus on deep thinking. For starters, stop multitasking.
Science shows us that multitasking is bad for your brain. It reduces fluid intelligence, causes brain atrophy, and increases chronic stress.
The global pandemic is creating more stress than ever, stress that leads to depression and anxiety and beyond. Unlocking our potential to navigate these hurdles starts with learning the right strategies, even in school.
So when teachers have these strategies, they're empowered to support our learners, and then the learners are now able to take ownership of their learning.
Training kids how to think is doubling academic achievement among middle schoolers.
I think the greatest national security threat is pre-K through 12. If we don't take care of educating our young men and women, then we have to ask ourselves, where are we going to be in 20 years?
Our world-renowned scientists know you can increase your brain health, not lose it. It's time for a new category of health, brain health. You are a game changer. Ready to transform the world with us? Be a part of the brain health revolution. It starts here.
All right, tonight's event is all virtual and free, along with the other 3 talks in this 2021 series. This is made possible by generous philanthropic support. And at this time, I'd like to acknowledge this evening's sponsor, Caliber Home Loans, and invite Rob Cushman to say a few words. Rob?
Good evening, everyone, and thank you so much for having us as a sponsor. I'm head of marketing and communications at Caliber Home Loans, and it's a pleasure to be sponsoring this lecture. Over the past 2 years, we've had a very close partnership with the center, And I'm proud to say that we're one of the earliest corporate supporters, I think, of this mission. And from our CEO down to many of our senior executives, we've incorporated many of the founding principles into how we conduct business. In fact, all of our most senior executives and a large number of their staff have been exposed to the most important facets of the program, such as the importance of brain health, what we can do to foster a healthier brain, and including a number of good daily principles, such as, I think was mentioned, focusing on the task at hand, not being swept into multitasking. That's resulted for us in something called elephant focus, which made us all smile when we first heard about it. And that was really a focus on the biggest, most complex project you had during the day. It also included things like, meeting etiquette, doing the right things for meetings, and perhaps most importantly, mindfulness. However each of us defines it, it's a way for us to recharge and focus. So really for us at Caliber, it's been a learning experience. We challenge ourselves daily to completely focus on these critical learnings, but we're committed to becoming known for the support of brain health. And in fact, we think that something like a brain health designation is something that will set us apart in our industry. So for us to sponsor this lecture tonight was a perfect fit into our goals and strategies. I'd like to just take a very brief moment to say thank you to Dr. Chapman for your continued partnership, and to Steve White and Jennifer and the entire team for helping us move ahead. We are absolutely delighted to be sponsoring tonight. And for everybody in Texas, Oklahoma, and all other surrounding areas that are affected by this horrible weather, I hope you're safe and warm. Thanks.
Thanks so much, Rob. That is a good point to remind everyone about. We are broadcasting out of Dallas, Texas, where we've had a lot of snow and power outages. So hopefully things go smoothly. They have so far, but just be aware that's likely the case if we do have any glitches. Okay, it's my pleasure tonight to introduce Dr. Nancy Kanwisher. Nancy is the Walter A. Rosenblith Professor at MIT and a founding member of the McGovern Institute. I first learned of her work on visual perception when I was in graduate school over 20 years ago. She was a rising star in the field who had worked with the legendary Anne Treisman at Berkeley and was now a pioneer in a new field of functional brain imaging. Dr. Kanwisher made very early and profound contributions to this area, exploring the links between brain activity and how we perceive the world around us. And for the past 25 years, she's established herself as a true leader in the field, connecting brain function to our subjective perception. This has included exploring how we process faces, objects, visual scenes, speech, and even our ability to anticipate the thoughts of other people. She is a member of both the American Academy of Arts and Sciences and the National Academy of Sciences. And Nancy hosts a website, Nancy's Brain Talks, which you can go to at nancysbraintalks, all one word, .mit.edu, which is another excellent resource for more fascinating information about the mind and brain. On a personal note, when I first started conducting functional brain imaging experiments of my own, the first meaningful result I found in one of my subjects was identifying the face and scene regions that Nancy and her colleagues had first reported on back in the 1990s. And I recall that thrilling experience of being able to actually connect a percept to brain activity. And it's just really profound. And many of Nancy's research findings have had a reverberating impact on many of us in the field for multiple decades. She is an absolute star in the research community. Okay, we're about to get going. Just a couple reminders before we start. Please put your view on speaker view setting for best viewing. And please use the Q&A box at any point throughout the talk. We'll have a little bit of time at the end for Dr. Kanwisher to address some of these questions. This event is being recorded, and it'll be shared with all the registrants. And now, without further ado, we bring you Nancy Kanwisher.
Thank you so much, Dan, for that really kind introduction. Well, hi, everybody. It's a real pleasure to be here. I hope you're all staying warm and safe. Up here in Massachusetts, it was 53 degrees today, which is completely unheard of for February, but there you go. I guess it means we all have to stop burning fossil fuels to get this craziness to stop. But I want to talk about something more fun today, and that is that I'm going to start with some good news, which is that our field of cognitive neuroscience has made a lot of progress in just the last few decades. And to get a feel for that, let me show you the approximate state of what we knew about the organization of the human brain circa 1990. we knew the very approximate locations of brain regions that were involved in perceiving faces, in paying attention, and understanding language from studies of people who had the great misfortune of having damage to those parts of their brain and losing those functions. So we had this very vague general picture with just a few kind of functions mapped on the brain to very big regions. That was kind of it. But then functional MRI came along, and here's where we are today. So there are now dozens of regions of the cortex in humans. That's the outer sheet of the brain. It's about the size of a large pizza. To fit it into your skull, you have to fold it up, stick it in there. So that's why the brain has all those wrinkles, so that all that surface area can fit in the brain. And so that cortex, we now know about several dozen regions in the cortex with a pretty good idea of what each of those regions does. And each of these regions shown here is present in approximately the same location in pretty much every normal person. But this is a highly schematic diagram here. Of course, we don't have neat little colored ovals in the brain. So let me tell you a little more about what the data actually look like that lead to this picture. So first of all, to do functional MRI, we use basically a regular old MRI machine of the kind that there are thousands, probably tens of thousands in hospitals all around the world. But there's two special things about functional MRI. One is that some very smart physicists, not me, figured out how to get these machines to show not just structure, but function. And second, how to take pictures really fast. So instead of taking, say, 45 minutes to get a series of pictures through your knee, they could take a whole series of pictures through the brain in a second or two. And further, those pictures show not just the density of tissue and the bumps and the folds, but the activity in the brain. And so if you put those two things together, that means we can make movies of brain activity. And the big hoopla about functional MRI has been because it gives us the sharpest pictures of activity in the human brain without opening the skull, which most people are not willing to volunteer for. So how can we use this method to reveal that map of brain functions that I showed you before? Well, let me show you some real data. This is one of my members of my lab, his very nice brain. This brain here has been mathematically inflated so we can see that whole cortical surface. And we're looking up at the bottom of the brain here. So the back of the left and right hemispheres are out on the side of the slides here, and they're kind of facing each other. So this is the bottom of the brain looking up like this, just to get you oriented. Okay, so the dark bits are the bits that were inside a fold before the cortex was mathematically inflated. Okay, and the light gray bits were out on a bump called a gyrus. So what are the colored bits? Well, that little blue blob there is telling us that that part of the brain responds more when you look at faces. That's the blue bar. This is the amount of neural activity in that region when you look at faces, compared to when you look at a whole bunch of other kinds of things. Scenes in green, objects in gray, and other kinds of images. So that region seems particularly interested in faces. And right next door, as Dan mentioned, is another region that responds really strongly when you look at pictures of scenes. And it doesn't matter what they are. It can be an indoor picture like your kitchen, or an outdoor scene of a forest, or the Southwest, a big open scene. Any kind of image where you look at that and you say that's a place will strongly drive this region here compared to faces, which produce no response at all, and objects and other things. Over in the left hemisphere is a particularly interesting little patch of brain. It's sometimes called the visual word form area because it responds very strongly when you look at images of words and letter strings. That's the yellow bar here compared to all these other kinds of images, faces and scenes. It doesn't like them at all. And this region is particularly interesting because it's the one part of the brain where we know that that selective response here is due to that person's experience, not something inherited from their ancestors. And we know that because you don't see this selective response to words until people learn to read. Before that, it's not there. And then after they learn to read around age 7 or 8, you can find it in every kid who's learned to read. And further, our species has only been reading for a few thousand years, and that's not enough for evolution to have built in through natural selection a special circuit for reading. So that one is trained up by experience. Out on the lateral surface, on the sides of the brain, we have some more good stuff, like these little purple patches there that respond strongly when you look at images of bodies and body parts, hands, knees, feet, torsos. They all drive this region much more strongly than faces or objects or scenes. So those are all aspects of high-level vision, but actually over here this is the temporal lobe here, that little orange patch responds selectively. It's responding right now in your brain. It responds whenever you hear people speaking. And the interesting thing about that region is it's not about understanding language. It's about hearing the sounds of speech. So if I suddenly switched into a foreign language you didn't understand and kept talking, that part of your brain would keep responding because it would detect that that's speech, okay? So this raises a question of whether we only have these specialized bits of brain for aspects of perception, vision and hearing and other things, or whether we might have them even for very complex, abstract, high-level aspects of thought. So a few years ago, my then postdoc, Evelina Fedorenko, decided to tackle the long debated case in our field of language. And so it had been known for at least 200 years that patches out here in red on the sides of the temporal lobe and the sides of the frontal lobe, especially in the left hemisphere, respond strongly when you're understanding language. But what Eva asked was, do those regions really respond only to language, or do they do lots of other things? This had been a big debate in the field, with many people arguing that language shares machinery in the brain with other things like music, like doing math, like just holding information in working memory, and lots of other things. So Ev developed methods to find the exact location of those regions in each individual subject. And then when she'd done that, we could then measure the response in those regions when people did a whole bunch of other tasks, music and math and working memory tasks, and see how strong the response was there. And so here's what she finds. This is a language region, sometimes called Wernicke's area. Here's its response in the black bar when you're understanding sentences. Here's its response when you're looking at a bunch of strings of letters that don't have any meaning. And here's its response, basically a big bunch of nothing, in all those other tasks. So this region responds really only when you're understanding sentences. Up here, this is a region sometimes called Broca's area, another language region, and it does the same thing. It responds strongly when you're hearing sentences and much less strongly in all of these other conditions. So what I like about this is it's not just another specialized patch of the brain, much as I think it's fun to find specialized patches of the brain. This is one that tells us something really deep and interesting. It tells us that language and thought are not the same thing. In the brain, they activate really very different regions of the brain. And that tells us that questions probably every one of you has wondered for a long time is, do we think in language? Could we think if we didn't have language? No, language and thought are very different. In fact, even stronger evidence for this comes not from our data, but from the studies of Rosemary Varley in England, who's been studying patients who have the great misfortune of having big lesions, big strokes in their left hemisphere that really knock out pretty much all of their language abilities. A very horrible thing. But what's remarkable is she's figured out smart ways to ask these people all kinds of questions. And it turns out that pretty much every mental function that you might have thought would require language doesn't. So people who can't speak or understand language at all can do arithmetic problems, they can do logic problems, they can appreciate music, they can hold information in memory, and they can think about each other's thoughts. So that's pretty even stronger evidence that thought and language are different. But perhaps my favorite patch of brain has been studied most extensively by my friend and colleague, Rebecca Sachs. And that's this little pink patch here. And this region responds only when you think about what another person is thinking. And if that strikes you as crazy, it did to me at first too, reflect on the fact that we humans do this all the time. Whenever you're talking to somebody, you're thinking about, what do they already know? Do I need to tell them this? What does that reaction mean? What did they think when I said that? We are just constantly thinking about each other's thoughts. It's pretty much what it means to be a human being, is to be spending a lot of your waking moments thinking about other people's thoughts. And so it turns out we have a special patch of brain just for that. And what's remarkable about Rebecca's work on this region is how strongly she's shown how very, very selective this region is. It doesn't just respond when you think about other people. If you're thinking about what they look like, it's not active at all. Even if you're thinking about their bodily sensations like thirst or hunger, it's not active. It's only active when you think about the content of their thoughts. So I want to hasten to add, before you all think I'm completely nuts, that I don't think every patch of brain is as specific as the ones that I just described. In fact, these white patches out there are part of a broad set of regions that go by many different names: the frontoparietal attention network, I call it the multiple demand system, because it responds whenever you do anything hard. It doesn't care what that hard thing is. It can be a difficult perceptual task, trying to see something difficult, trying to hear something difficult, trying to solve a math problem, just trying to think hard about anything difficult. Those regions turn on. And so they're sort of like the opposite of all the other regions I've described. They're not specialized for one particular thing. They're just like, it's like the brain's CPU or something. When you have to think really hard, they step up. Okay, so that's my whirlwind overview of some of the cool patches of the cortex that my lab and other labs have been studying over the last couple of decades. But what I want to do then is step back and say, I think this is real progress, that we now have this picture of the brain, a partial picture, but a pretty cool one nonetheless. It shows a set of functionally very distinct regions that are present in pretty much the same place in everybody. And that includes regions that are specialized for very abstract, uniquely human functions, like understanding language and thinking about other people's thoughts. I like to think of this as an initial sketch of who we are. That this is our machinery. This is what it is to be a human being. Let's have this stuff running in your head. But at the same time, this is the barest beginning. We have barely scratched the surface in understanding the brain and how it works. And really, I think the way to think about this diagram is as a roadmap for future research. And so what I want to do in the rest of this talk is discuss some of the kind of wide open questions about the brain that are just really raised just by looking at this picture. You have to wonder these questions. So the first one I'll tackle is, what is the causal role of each of these regions in perception and thought? What do I mean by that? What I mean is, with functional MRI, you put a person in the scanner and you show them some pictures or ask them to do a task, and you see some part of the brain turns on. It doesn't tell you that that region is necessary for that task. It just tells you it turned on when people did that task, okay? But we really want to know about causality. Science is not about what tends to happen with what other things. It's about what causes what. And so this really matters in science. And the sad thing is, functional MRI is not going to answer this question. So that's a big bummer, but it's just reality. And so we face it and come up with other methods. And that's why I was really thrilled when a few years ago, I got a phone call from a colleague telling me that this gentleman was a neurosurgery patient at that moment in a hospital in Japan. And he was having his brain mapped out before neurosurgery for intractable epilepsy. So most people with epilepsy, it's treated well by drugs, but for some people, it's just not responsive. And if the seizures are frequent and severe enough, you just can't live a normal life. And in that situation, many people actually have a real improvement in their quality of life if neurosurgeons go in and remove the focus of the seizure. So this gentleman was in the hospital, and here's a picture of the bottom of his brain where the neurosurgeons had placed electrodes all over the bottom of his brain to map out different mental functions and to find the source of his seizure. And so my colleagues asked if I wanted to collaborate by sending them some images so that they could record activity in this guy's brain while he looked at those images. And I'm forever grateful to him for agreeing to look at my stupid pictures so that I could measure responses in his brain. And for comparison about why those electrode locations were so exciting to me, here's a picture of the bottom of my brain. The red bits are my face areas. The green bits are my place areas, mostly up in that fold, so you can't see much. And the purple bits are bits that respond more when you look at things in color versus grayscale. So it looks like the electrodes are right over some of the good stuff in his brain. So we shipped some images over the internet to Japan, and the neurosurgeons showed him images of all kinds of different things, pictures of bodies and digits and Japanese characters and line drawings of objects and faces. And they sent me the data showing the response of each of those little electrodes in his brain. So for example, right here, right on top of what looks like my face area, is an electrode, and here's its response. This is time on the x-axis, 2 seconds here. And you can see, and this is the amount of neural activity on the y-axis. So you can see this huge burst of activity only when he looked at faces, not when he looked at any of these other kinds of stimuli. So that shows you the remarkable selectivity of that region, much stronger than those bar charts I showed you from functional MRI. And that's because we have much higher resolution with this method than we do with functional MRI. So far, that's just showing that we validated the results of functional MRI with a method that's kind of closer to the source, actually recording electrical activity from neurons right on the surface of the brain. But the question I raised at the beginning is, what is the causal role of that region? And that's why it was particularly interesting to me scientifically that the neurosurgeons had decided to electrically stimulate some of those electrodes. They do that as part of their method for discovering the seizure focus. So I'm going to show you a videotape of this gentleman being stimulated right there on top of those very face selective electrodes. And he's simply asked to report what he sees and if anything changes. So here we go. The sound is quiet and it's in Japanese anyway, so read the subtitles. And recall, this guy has no idea. So here he is showing that that electrode's being stimulated. He has no idea where he's being stimulated, right? So that shows that that region is causally involved in face perception, because when you stimulate there, it changes the percept of the face. Here we're asking, is it causally involved in other things? When he's looking at a box, what happens?
No change. 最初の1秒ぐらい、始まって1秒ぐらいがなんか横になんか目口って感じでちょっと一瞬見えたんですけど、なんかなと思ってたら気が付いたらもうこの箱にしか見えてないっていうか。なぜかまたさっきの横に。目と口って感じでちょっと。
So this is a Japanese character on a card here.
More, please. One more time.
So you get the idea. What it shows is that when you stimulate that very patch of brain that responded selectively to faces, he sees a face. If he's looking at a face, it changes the face. If he's looking at something that isn't a face, he suddenly sees a face. That's causal evidence. That tells you that it's not just that that region responds when you see a face, that region is actually causally involved in your percept of a face. So, oops, here we go. So what is the causal role? Well, this is the strongest kind of evidence we have for the causal role of that region in face perception. And I have another video. You can ask me if you want to see it later, but I'll just describe when he's stimulated right next door in that little patch that respond more to colored than grayscale images, he says, "I see a rainbow," which is just incredible, but that's what he says. So that tells us that that at least these regions, we have very strong evidence for their causal role in perception. Okay, next question. Obvious question you've probably all been wondering already. Okay, we got some interesting stuff here, but what else? What other kind of crazy stuff is there in the brain? And of course, in my lab and lots of labs, we've been asking that question. And I'll just mention two recent lines of work. One is this little patch right here, which seems to be involved not just in perceiving other people or thinking about what they're thinking, but in perceiving two other people interacting with each other and asking yourself what kind of interaction they're having. So we can actually decode from the pattern of neural activity right in that little blue patch, whether the person is looking at two people having a positive interaction or a negative interaction. So we think that's actually a really important aspect of social perception, is perceiving not just single other people we may be interacting with, but pairs of other people interacting with each other. Another set of regions we're investigating is these ones in purple, and we think they are involved in actually objects interacting with each other in physical ways. And if you think about it, a basic understanding of intuitive physics is actually essential for any navigating organism like us. You can't pick up an object without first having some kind of idea of how much it weighs and what the friction is of its surface and if it's rigid or squishy or what. You can't walk any place without first deciding whether the floor will support your weight and whether you can go through this barrier or not. And so we have some evidence, it's early days, but we have some evidence that these brain regions are involved in this kind of reasoning about the physical world. But what I want to mention in a little more detail is some work where we've been looking at auditory cortex. And this is really exciting. We've taken a very different kind of approach from the experiments I've described so far, where instead of coming up with a hypothesis like, okay, maybe seeing social interactions is important, let's test it and find out. And sometimes you get lucky and sometimes you don't. With auditory cortex, we said, okay, let's scan people. Well, they just listen to a whole swath of different common everyday sounds. So people talking, toilets flushing, ambulance sirens, dogs barking, you name it, just common everyday sounds. And we record the activity in the brain while they do that. And then we do some math that I won't go into in detail, but it's basically a way of taking those data and saying, okay, show me the simplest version of the structure in these data, okay? And so what falls out of the math is something really astonishing. It discovers some basic components, which you can think of as sets of neurons that respond the same way. So here's one component it discovers. This is the magnitude of response to each of those 165 sounds, and we've color-coded them by the category of sound. So if we then average within category, what we see is that this set of neurons responds whenever you hear speech, and it doesn't care if it's foreign speech or English speech. The blue bars are music with vocals. So that also has speech, although somewhat muffled, harder to hear. So this is a speech selective response, okay? And so this was cool. It wasn't entirely new. People had reported stuff like this before, but with our 165 sounds, we have really strong evidence about how very, very selective this region is. But the other component we found was really brand new and quite astonishing. And here it is here. If we average within categories, we see this set of neurons respond strongly when you listen to music, instrumental music or music with vocals, compared to all these other categories of sound. And that was just astonishing and new to us. And further, it answers a question that people have wondered about for a long time. Nobody knows. Darwin wondered why humans have music. Didn't make sense, right? Being able to perceive and speak to each other with language makes evolutionary sense. You need it to survive. But what is the survival value of music? Nobody knows. And so one theory had been that music is kind of a byproduct of speech. Once we have neural mechanisms that enable you to speak, you can co-opt them and get them to do other things. And our data say, nope, that's not right. So these neurons respond strongly to music. They're really not interested in speech. These ones do the opposite. There's a total separation in the brain. So whenever you see something really astonishing like that, one's response, at least my first response is, really? So we were thrilled when we got an opportunity a little bit later to get more recordings from neurosurgery patients who had electrodes on the surface of their brain. And so what we saw is some neurons, some electrodes that responded very selectively for speech. This is now time here. The two green bars are speech. Others that respond selectively to music, whether vocal or instrumental, validating thus the responses that we'd seen with functional MRI. And others that respond only to vocal music. And that was a total surprise, that we'd have special groups of neurons in the brain that aren't just the sum of a response to speech and a response to music. No, they respond only to vocal music, not to speech, not to instrumental music. So that was pretty exciting. So we get to add more blobs on the brain, and we're still at it. But I want to discuss, I'm going to skip a few little details here. I want to ask a big question, which is, how do you wire up a brain to have all this intricate structure that's so similar in each person? How does that work? I'm not going to answer that question, but I'll mention a couple of interesting results. So a number of studies had argued that that face area develops really, really slowly, and it's still developing right through adolescence. Okay. But on the other hand, there's another study where they raised baby monkeys without ever letting them see other monkeys. They heard other monkeys. They had lots of cuddling by human caregivers who had masks over their faces, but they never saw faces. And they did not develop face patches like normal monkeys do. I forgot to say monkeys have these same face patches that humans do, but not if they're raised without ever seeing faces. So that suggested that to build a face area in development, one, it takes many years, and two, it requires visual experience with faces. So we asked, really? And we asked really a couple of ways. One is the prior functional MRI studies of development were all on older kids, age 4 and up, but that's too old. We really wanted to know what happens with infants. And scanning infants is almost impossible. But my very heroic friend and colleague, Rebecca Sachs, spent 5 years figuring out how to scan infants. One of the great lengths she went to in this research enterprise is to produce her own subjects. This is her son, Arthur, who's probably been scanned more than any other 6-month-old infant. And this is her grad student, Ben Dean. And this is some of the beautiful data they got, showing that those face and scene responses are present already in 6-month-old infants. But this work doesn't yet tell you that those regions are as selectively responsive as they are in adults. It was kind of hard to tell with infant data that infants move and squirm and you don't get much data and it's hard to tell. But so enter the scene the amazing Heather Kosakowski, who's a grad student with me and Rebecca Sachs. And she is probably like the world expert on scanning infants. Heather can take a screaming, squawking, kicking infant and somehow get beautiful MRI data out of them. It's a miracle. And so Heather has found face, place, and body areas in 6-month-old infants. And she has further shown that if you measure the response of those regions, they're really, really selective. So here's the face area responding to faces versus other things, the place area responding more to places and other things, and the body area responding more to bodies and other things, showing that face, place, and body areas are actually present and with nearly adult-like selectivity in 6-month-old infants. I'm going to talk about one more experiment and then I'm supposed to stop, but I got to tell you one more experiment. This says, no, it doesn't take years to develop. But what about that question? Does it require visual experience? So do we need to see faces to develop face areas? Well, one way to answer that is to scan congenitally blind people. So we did that. We brought congenitally blind people into the lab and we had them feel 3D printed objects that were either little heads or mazes or hands or chairs. So the person is lying in the scanner. Here's the scanner bore. And there's a disc with these objects glued on it, and they're feeling them as the disc is rotated while we scan their brains. And when we do that, first with sighted people, Looking at these images, we see this face selective response with a high response to faces compared to other things. That's expected. But what do we see with blind subjects? We see pretty much the same thing when they feel faces. The overall response magnitude is lower. The numbers are kind of hard to see here, but the pattern is very, very similar. So that tells us that no, you don't need to see faces to develop a face area. So we don't exactly know how these things arise in development, but we know they arise really early, and that there's something about that patch of cortex that is marked to be a face patch, even if you never see faces. So I will just mention the other wide open questions without going into any detail. Of course, we want to know, how does all this structure arise over evolution? And we can compare human brains and monkey brains and see what's similar and different. Bottom line, visual cortex, very similar. Auditory cortex, really different. And so there's lots and lots of other questions. Ultimately, we want to know in detail what's represented in each of these regions and what computations are going on in there. We now have computational models that enable us to actually make guesses about how the brain computes. We want to know how these regions are connected to each other and to the rest of the brain. That's a really wide open and important question. And of course, we want to know, how do these regions interact to produce intelligent cognition and behavior? And to me, the biggest question, which we're actually working on in my lab right now, is why is this a good way to design a brain in the first place? To have all these special purpose bits doing different things. Why not just have a general purpose brain that's really smart? It can handle anything at all. And I think that's now very recently become a tractable scientific question. And I will just leave this roadmap up there, mostly questions, not answers, and say thank you. And I'm happy to take your questions.
All right, fantastic, Nancy. So interesting to consider what's going on up there when we have all these different thoughts. We've had some great questions coming in from the Q&A. I'll get us started with one about autism spectrum disorders. So people who have an autism spectrum disorder, the question is, are these areas for perception physically or neurologically different, given that those people sometimes seem to miss information about other people's minds?
Yeah, it's a great question. And it's one we've done a little bit of work on. It's a tough question because, of course, there's a huge spectrum of the degree to which people with autism can be affected. And of course, it's not one spectrum, it's many. There are many different heterogeneous manifestations of autism. But people with severe autism are very hard to scan because, scanning's not for everyone. I love it in there. I'll go in there and hold still for hours. I'm so excited about the data I'm getting. I don't mind it at all. But it's tough. And so mostly the data we have is from very high functioning autistic people who kindly go in and let us scan them. And so the first surprise is that yellow patch, which has somehow disappeared in my this image, I don't know why, but anyway, there's that yellow patch there, the region that it was pink when I showed it to you before, the region that's engaged when people think about each other's thoughts. That would be your first target area. Like, would that be affected in people with autism? So Rebecca Sachs has shown that surprisingly, in high functioning people with autism, it's not affected. It's the same size, it's in the same place, it responds with the same strength and the same selectivity. And so that's kind of surprising because you might think that would be the first thing to be affected. Rebecca has found some very, very subtle differences that if you look at the pattern of response across that region, in typical people, you can read out, you can kind of decode from the brain whether people are thinking about another person's false or true beliefs, or you can tell some other things about the nature of the other person's belief they're thinking about. And that she was unable to do in people with autism. So it suggests that even though the region is there, it may not represent all the same information. There's also a lot of work on the face patches in people with autism. And it seems, although there were some early reports for around a decade where people thought those face patches were really different, in our hands and in lots of other labs, actually, mostly the whole face system seems to, at least it's there as far as you can tell with functional MRI. It's there, it's in the same place, it looks really similar. So that's pretty unsatisfying because obviously something's different, but it's not in the basic core components of the brain that are all there in the same place, doing more or less the same thing. It must be something subtle about actually how they work.
Fascinating. Okay, another question about meditation. So do we use the same regions when meditating or visualizing something within our minds, maybe with our eyes closed? And what about when we intentionally manipulate a scene or something we imagine?
Yes. So yeah, we showed one of the first experiments I did right after we found the face and place areas is I popped a colleague in the scanner and I said, okay, close your eyes and I'm going to shout out some names of famous people. We had very rudimentary methods back then. It was basically me screaming into a microphone and she's in the scanner like, what? It's like, and the instruction was form a vivid mental image of each person or place. And indeed, when you imagine faces, you selectively turn on your face area. When you imagine places, you selectively turn on your place area. And I love that because, you know, we humans are very, very visual animals. Basically, the whole back third of the cortex is involved in different aspects of vision. And you might think, is it really that important to see? And well, it's pretty important to see. But what's cool is we can use all that same machinery to think with. We use it in mental imagery. We use it in all kinds of ways when we, imagine, diagrams. We think very visually. We use a lot of the same machinery when we do that.
Yeah, such a deep question there. It's nice to know our thoughts are real at the level of the brain as well. Another interesting, fascinating kind of question related to that brain stimulation clip that you had shown. Does your research tell us anything about how we might stimulate the brain for people who suffer from impairments to improve their thinking?
Yep. This isn't something I work on, but a number of people do work on brain-machine interfaces where you basically have electrodes on the surface of the brain, like in that patient, where you can record information and then you can use it, for example, to direct a robotic arm in a paralyzed person. And that, they don't work great yet, but there's research going on improving them and they are getting better. And I just think that's magical and thrilling. When I was 15, I jumped off a friend's shoulders at the beach and broke my neck and was a quadriplegic for a while. And I got lucky. The surgeons patched me up and here I am and I'm fine. But I was quite paralyzed for months. And it's very vivid to me that there are people who didn't recover as luckily as I did and who may, in not too long, have various ways of being able to, basically think the will to move an arm and have that signal read out directing an arm. There are also some recent studies where people have been putting arrays of electrodes over visual cortex in the back of the head here. And actually a paper that just came out a few months ago from somebody in Texas, Michael Beauchamp, who had a set of electrodes over visual cortex. And they basically drew patterns on the cortex with electrical activity. And the person just drew on a piece of paper what they were seeing from that electrical activity. So that's super exciting and suggests that in people who are blind because of, impairment in their eyes, some people are blind because of impairments in the brain, but if your impairment is in your eyes, then you may be able to bypass that eventually by using a camera, translating it into electrical activity and just putting it straight on the brain. We're not there yet, but there's some exciting first steps.
Fascinating world ahead. A question that comes up, there are a lot of nature-nurture kinds of relationships throughout your talk. One of the intriguing ones is multilingual individuals. Have you studied people who are bilingual or trilingual, and do you see any reliable differences either in that visual word form area or maybe even within sort of the Broca's and Wernicke's areas?
Right. Well, I haven't, but my friend Ev Fedorenko, who did that work I described at the beginning of the talk, showing the selectivity of language regions, she's been studying this. In fact, there's a wonderful New Yorker article that came out around a year ago about her research. So she's been studying polyglots, these people who speak an insane number of languages, like 20. So like there's a number of these people who are just incredibly skilled at speaking different languages. And so she brought a number of these people into the lab and scanned them And what is her result? Her result is, first of all, when they hear all the different languages that they know, they activate the same parts of their brain, not different parts of their brain. All those languages are right on top of each other, and at least at the crude scale we can see with MRI. But second, the main difference you see in their activity when they listen to language is there's less activity than people who just speak one language, which is pretty wild, but there it is. And maybe you can argue maybe their system just becomes so efficient, it just doesn't require that many neurons firing. I don't know what the right answer is, but that's what she sees.
Fascinating. Another question comes in about emotion. So a lot of what you've talked about has probably some gateway components to how we actually feel our emotions. And what would you say about the role of emotion, maybe at the level of the brain? Does emotion have a region, or does it somehow get distributed in these regions?
Yeah, so the standard view is that the kind of core emotional functions are in subcortical regions. So I have a very corticocentric view. I've been just talking about that large pizza that's shoved into the brain, but of course, underneath all this cortex is a whole set of other regions that are involved in all kinds of really important functions, very importantly, including emotions. So some really well-known ones like the amygdala that plays a major role in fear, and other subcortical regions. That said, there's an emerging view that actually we don't just experience 5 or 6 emotions. We experience a very rich swath of subtly different emotions that have very different meaning from each other. And so the idea that's emerging is that very rich array of different emotions engages the cortex, because you have to put your interpretation of what the meaning of that situation is. And so that kind of cognitive overlay is part of the richness of emotional experience.
Okay, here's kind of a bit of a challenging question. Do you find that there are differences in these areas between men and women? And I guess that could be at multiple levels. Like, do most men and most women vary, or do you find maybe differences when viewing male or female humans?
Mm-hmm. Yeah, I used to get this question a lot, and it always made me nervous because I thought, if I ever found a sex difference in the brain, it would be reported in ways, to have significance that it didn't. And so for a long time, I didn't look because I just didn't want to go there. I didn't want to be responsible for people misunderstanding what any possible sex difference in the brain was. And then actually a colleague of mine made a very good argument. Actually, he was funding some of my research on autism. And he said, look, there's a huge sex ratio in autism. It is much, much more common in boys than girls. And we want to understand why. And so since I'm funding you, I'm asking you to go look at the differences between men and women in your face areas. And I thought, okay, that's a good argument. And I went and looked kind of grudgingly, and there was no difference whatsoever. So I haven't looked at every possible region of the brain, but of the ones that I've studied, there certainly isn't anything obvious. And, I think that, people think, each person thinks somewhat differently than the next person. But what this research shows is that we do that with basically the same components, right? We have the same basic parts of our mental machinery in the brain. And so there's going to be differences in how we use it, but they're pretty subtle, and they don't seem to segregate around male versus female in any major way, as far as I can tell.
Okay, that sounds like a fair answer. I wonder, there's a question, another practical one. Some people are good at recognizing faces or remembering what people look like. Others profess to have terrible memory for faces. Can you comment at all from your work, or maybe work of others that's related? Do we know anything about face memory and talent in that area of the brain?
Yep. Well, I would say the behavioral work is the most interesting here, just regular measuring people's behavior. And what you see is that there are indeed massive differences in people's face recognition ability. If you take 100 people, the 2 best ones out of the 100 are spectacularly good at face recognition. And the bottom 2 to 5 are really, really bad, like routinely failing to recognize family members. So just in any normal group of people, the breadth of the range of abilities is just enormous. So that's the first thing. The second thing is, I'm happy to say that your face recognition ability is not correlated with IQ, not one teeny bit. And that's nice for me because I'm not very good at face recognition, but it's also nice because it sort of fits with this picture of the brain. To the extent that we have separate machinery for different mental functions, why would how well this part of your brain works have anything to do with how well you think? It's just a separate part of the machine, right? The other thing I'd say is that behavioral studies in twins have shown that face recognition ability is highly heritable. And that's so if you have 2 identical twins, their face recognition ability will be more similar than if you have 2 fraternal twins. And that shows you that genes have something to do with it. But that doesn't mean necessarily that genes specify that that face area could be right there. Maybe what the genes do is make you, for example, more extroverted. And maybe if you're more extroverted, you look at faces more and you train yourself better to recognize faces. So heritability is funny. It could be, in that case, showing really an experiential effect in disguise. So I sort of went off the track. But anyway, yes, there's huge range. It's heritable. It's not related to other abilities. And so what does that have to do with the brain? So the weird thing is this face area down here is not radically different in people who, at least at the grain we can see with our current methods. It's got to be different somehow. But with the current methods, it looks really similar in people who are great at face recognition and people who are terrible. Maybe slightly smaller in people who are terrible, but it's not a big effect. And so that just tells us that there are things that our fabulous methods can't see.
There's always more to be discovered. This is a good question for you. You probably get this a number of times. Hemispheric differences in the brain. So left versus right side. You talked a little about speech and language having a left-sided focus. And some of your work with Rebecca Sachs emphasizes a right-sided focus for that perspective taking. And so just some curiosity, what would you say about the left-right organization of the brain? Are there any sort of like solid rules for it, or is it highly plastic like many things?
Well, so first of all, the old car commercial version of it, the artistic side and the logical side, that's a big bunch of bunk. But that said, there are clear differences. And as you say, the language is in most people more in the left hemisphere than the right. Face areas in most people are more in the right than the left. Many of these regions have one bias or the other. But the significance of that is really hard to figure out. Actually, you're reminding me the one finding that we and others have found with autism is actually you get less lateralization in people with autism. We don't know what that means, but they're less biased to one side versus the other. But the other thing I'll say is that if you have brain damage early in life, many of these regions can move over to the other hemisphere. So in the most extreme cases, really tragic cases of young kids who have very severe epilepsy, in the most extreme cases, they've sometimes surgically removed pretty much a whole hemisphere. And the amazing thing is those kids do really, really well. So you can take an entire left hemisphere in a, 2 or 3 year old, and by the time they're 10 or 12, their language is nearly normal. It just takes up residence in the other hemisphere. So there's a lot of plasticity early in life about which hemisphere those functions can land in. But at the same time, they're very systematic patterns of where they tend to land in most people.
Well, you'll be happy to know you're getting fan mail in the Q&A. One comment is, can I come live at your lab? I want to know more about how the brain works. It's just the kind of enthusiasm we hope people experience when they hear a great talk like this. I will finish with one last question, which I just have to ask, came in on the Q&A. What's your favorite part of the brain?
Oh, geez. I don't know. I guess I'd say my favorite part of the brain are bits that I don't yet understand, but have an idea how to study. And there's a number of those. We have new methods using computational tools that we think can tell us something about these regions down here that you notice I haven't colored in, That's not because they're not doing anything. It's because I don't know what they're doing, right? And so that to me is full of potential and excitement. And we now have new methods to go in and ask in a more sophisticated way what those regions are doing. So that's my favorite thing, is discovering new stuff in the brain.
A great sense of curiosity. Nancy, thank you so much for this remarkable talk, and thank all of you for attending. Let's go ahead and wrap things up. I just want to remind everyone that lifelong brain health is our shared mission. Thank you, Dr. Kanwisher, for your remarkable insights tonight. And we will remind our audience you'll be receiving a very brief survey that'll help us to improve our process for these talks. So please take a moment to complete it if you've enjoyed this talk. And we hope you come away inspired, remembering that your best brain years are ahead. And please consider joining the Brain Health Project at brainhealth.utdallas.edu. That's brainhealth, all one word, .utdallas.edu. Please tune in again next Tuesday to learn more about nutrition in the brain with Dr. Drew Ramsey. Thank you. Have a good evening.